irrigation.
Welcome and thank you all for standing by. Participants will be in a listen-only mode until the question and answer session. At that time, if you would like to ask a question, please press star then 1. Today's call is being recorded. If there are any objections, you may disconnect at this time. I would now like to turn the call over to Mr. David Lamb. Sir, you may begin.
Thank you. And I want to welcome everybody to the fourth webinar in the Mississippi River Basin Healthy Watershed Initiative, where we're focusing on those things that we can do to improve soil quality and have a positive impact on the nutrient loading that happens to be going down the Mississippi River. Today's topic, we're going to be talking about the role of soil biology in improving soil quality, and hopefully you all have received a copy of an email that has a little flyer like that in there, just for future reference. When you see that, you know there's a webinar coming up, and you can just say, click here to join the webinar, and that'll take you right in, and you walk through the, walk down the steps there, and you ought to be able to get into the webinar. All of them are at 2 o'clock Eastern Standard Time on Wednesday. Our next one will be on August the 10th. We have made arrangements to get CCA continuing education unit credits, but it's up to you to apply for them. What you have to do is go on right where, just follow the red arrow over there. On your top of your screen, you should see a handout button. If you mouse over that, it should have handouts show up. And when you click on it, you'll get a screen that looks like this that has a whole list of documents that Dr. Nichols has provided for you to download that will help further your understanding of soil biology and the work that she's been doing and the significance and the role that it plays in improving soil quality and nutrient availability and nutrient cycling. Also, you'll be able to click on there. You need to check it too. Put a little check mark in there if you want to download it, where it says documentation for certified crop advisors. You click on that and you download that, and then you're responsible to print your name, put your CCA number there, and then sign it, and you can get it back to here at the East National Technology Support Center in 2 different ways. One, you can fax it and make sure you put attention to Holly Kirkendall, or you can scan that document after you sign it and email it to holly.kirkendall@gnb.usda.gov. And you need to get that back within the next 2 or 3 days so we can process these. And then what we do is basically we look at what you send in, we link it, provide the CCA folks with a listing of printed, with those who have participated that we get from the webinar folks, and they can verify that you participated in the webinar. If you want to ask questions, there's 2 ways you can do it. We have all the phones are on mute right now, but at the end of the session, you can do it audibly. You can press *0, and you'll get the operator, and she'll introduce you. You can give her her name, and then she'll bring you in and allow you to ask your question that way. Or the other thing you can do is, again, at the top, you have a menu list there. You click on where it says Q&A. That stands for question and answers. You just type in your question, and it'll go into a queue, and then we'll answer those as they come in. So again, 2 ways. One, if you want us to hear your voice, or the second one, if you want to type in a question. That one might be kind of handy as you're going through. If Dr. Nichols brings something up, and midway through the presentation, you want to remember it, you can type your question in at any point during the presentation. But we will be only answering questions at the completion of the presentation. And now let me introduce Dr. Nichols. I'm really excited about this topic because I know all of us that meet the soil conservationist series qualification had a bunch of soils classes when we were back in college, and we all learned about the physical, chemical, and biological aspects of soil. But somehow or other in my career, when we checked out of soils, we checked out of the biological part, and we focused solely on the chemical and the physical. And somehow or other throughout my career and the way I was trained and brought up in the Midwest, the biological aspects was never emphasized. And I think that's really been a key to this whole issue of soil biology. We're talking about increasing organic matter, is understanding the important role that the biology of the soil has in this, and how its significance in maintaining sustainable soils for sustainable agriculture. But anyway, Dr. Nichols was raised in Minnesota basically on a corn soybean farm. She received her Bachelor of Science degree from the University of Minnesota. So she's in the Big Ten there. She got her master's from West Virginia University, which she shows how daring she is. She was telling me earlier how she went there sight unseen to get educated in microbiology out there. She earned her Ph.D. from the University of Maryland. And she's been on the cutting edge of this whole idea of soil biology with and I'm not even going to try and pronounce those words, I'll let her do that, with the plant root system. I'm involved with investigations on glomalin, and you can read the rest. But I think we have probably one of the leading speakers on this topic that we could have got, so I hope you sit back and enjoy this. And Chris, I'm going to turn it over to you, and we'll go up here and bring up your presentation.
Thanks a lot, David, and thank you all for attending and for the committee inviting me to give this presentation. I'm really excited about having this opportunity. I love to talk about soils, so you may end up having to cut me off at some point in time and just say, okay, that's enough, because I could talk about soils for hours, and especially looking at soil biology. I think David made a very good point in the fact that a lot of times through our soils education, we learn a lot about the physical and the chemical sides of soils, and that's a lot of the analyses that we do when we look at soils. But often we don't look that much at the soil biology and the interaction that that has. And so hopefully I'll give you some ideas on the role of some of the organisms that are in the soil, and what you can do to help manage the system so that you're able to get more of these organisms in your soils, and what some of those impacts are going to have on kind of the whole system. When I was first asked to do this, I sort of started off and I was trying to focus on the Mississippi River area, and then I realized that a lot of these concepts are very general. They're going to be some things that I believe, when you focus on soil biology, you may be adjusting or doing some of the methods a little bit differently, or utilizing some different crops, but the concept is the same. It's going to work wherever you are. A lot of times I've been in various areas of the country talking to farmers, and a lot of times they'll say, you know, I hear all of these excuses why your system doesn't work. It's too cold, it's too hot, it's too wet, it's too dry. You know, it's never going to be the perfect season. As David said, I was raised on a corn soybean farm, and I told my dad that if he ever had the perfect season, he'd probably die of a heart attack, because it's never going to be perfect. But what we're trying to do is we're trying to come up with a system that's going to help you manage whatever it is that you have and whatever conditions occur. When you focus on the soil biology, you can see by the huge diversity of numbers and diversity of activity that you have a lot of redundancy of function, which means that whether it's too cold or too hot or too dry or too wet, there are going to be some organisms in the soil that are going to be working, and they're going to be able to provide some of these benefits to the environment that you're growing your plants in. And so, you know, the quote on the bottom, I really believe in, that civilization sort of rests on the soil, and they survive as long as the soil is not exhausted. And we're, I think, rapidly heading to a point where there's going to be a lot of issues with the soil, and hopefully we can stop it so that the soil is not completely exhausted, because we need to maintain that resource. I believe it's the most precious resource we have. We can, you know, we're talking a lot these days about energy and things like that, and we can find other sources to produce energy. We can find other sources to alleviate some of the depletions that we have in some of our resources. But the soil resource is not a resource that we can find alternatives for. We can't find the alternatives that are going to allow us to be able to produce enough food to feed our growing population. So, you know, I just kind of want to set the stage with that. I reviewed a couple of the earlier speakers, and so a lot of this information you probably heard from some of the other speakers. I'm going to try not to be too redundant, but also kind of emphasize, again, some of the important topics that exist out there with soil biology. So to begin with, I want you to think of the soil as the heart of the system. It basically is what's going to be connecting above ground and the below ground activity. So the organisms that are above ground and the organisms that are below ground are connected via the soil environment. It's also the ultimate recycler of primarily carbon and nitrogen and oxygen, but of a number of different elements as well that are necessary for the plants to be able to grow. It drives the physical and the chemical and the biological processes that hold the system all together. And it is estimated that the value of the services provided by the soils, that includes soil acting as a foundation for building, soil for growing crops, the biological activity, the organisms from the soil, is over $20 trillion on a global basis per year. But just the activity of the organisms themselves in the soil, fixing nitrogen, cycling phosphorus, providing the nutrients to the plants from that soil environment, those organisms are valuable, extremely valuable, and it's estimated that their value can be well over $1.5 trillion. So we're looking at a resource that definitely has a very strong economic value, not just a social and environmental value. And the biggest part of the soil, and the thing that we can see the most and identify what makes a good soil versus a bad soil, is that dark, rich organic matter that may be in the soil. And it's really a very small amount of the total soil weight, but this is where almost all of the functions that occur in the soil happen. That organic matter is primarily negatively charged, but it binds both cations and anions. And as you can see from the figure below, you have this activity that's moving, these nutrients that are moving from soil colloids into the soil solution and then into the plant roots. And a lot of that activity is driven by the root exudates, which are feeding the organisms that are in the soil. So you have nutrients that bind chemically with the soil minerals, but then you have mechanisms via the soil organisms to release those nutrients and provide them for the plant growth. So soil organic matter, as soil organic matter increases, you can also increase the amount of water holding capacity. The organic matter, like I said, it's a very small amount of the weight of the soil, but it's a large amount of the volume. And that volume versus weight allows it to act like a sponge, and it will be able to absorb water. And it's an area that stockpiles a huge amount of the amount of carbon that is actually in the atmosphere of planet Earth. So it's this huge stockpile and the storage of carbon. And that carbon is very important. This organic matter is very important, and the carbon that's in it is very important, because it's the primary food source, either directly or indirectly, for most of the organisms in the soil. And when you're looking at trying to grow and maintain those organisms, the two things I want you to keep in mind for this presentation and the things we're focusing on are the basic needs of the organisms in the soil. And that the first basic need, just like any other organism that these soil critters have, is food. And then the secondary need is habitat. And so what we're trying to do is we're trying to create an environment in the soil that has the maximum amount of food and the best habitat to maintain these organisms, so we can start seeing the functions increase and improve in our soil systems and improve soil quality. This is sort of a rough diagram of, again, the concept of soil organic matter and how much soil organic matter there is. Again, like I said, the soil organic matter can be 1 to 6% of the amount of the total soil mass, and that organic matter is primarily composed of the stable fraction, the humus fraction, that gives that dark richness to the soil. And then you have the more readily decomposable, the plant material, those types of things that are going to be in soil environment. And a smaller fraction of that is actually the organisms themselves. The bodies of the organisms are considered part of the total soil organic matter. The most dominant as far as weight is concerned organism is the soil fungi. And then the most dominant as far as the number of individual organisms is actually the bacteria. So in this case, we're also looking at parameters that involve size. The reason that the amounts are divided up the way they are is not just because of the numbers of the organisms, but also how big they are. So you may have very few animals in the soil, the microarthropods, and algae and protozoa and organisms like that, but they actually can make up quite a bit of the soil mass because of their large size. Now, in looking at the soil rhizosphere, this is the zone that is right around the plant roots. And I think that we're going to see more and more that basically I'd sort of phrase it as this, that the root of the problem is the root of the solution. If we have soil roots in our environment, it's going to solve the problems that we have in production agriculture. And a lot of times we look at various problems and we're actually not looking at the problems. We call them problems, but they're actually symptoms of a greater problem. And that greater problem is a lack of carbon and a lack of microbial activity in the soils. Most of the carbon that's made by the plant during the growing season is actually transmitted into the roots and then out into the soil to feed those organisms that are going to be living around the rhizosphere. So having a living root growing in that environment can have a really big impact on the soil environment itself. It impacts the soil structure, it impacts nutrient cycling activity, and this environment is where those organisms are directly connected with above-ground management. So even though these tiny organisms may not have any contact with the soil surface, they are definitely impacted by that environment through how we manage systems and how that impacts the roots and the amount of carbon that's given to the soil environment through the roots. The soil livestock is a very diverse, it's a very integrated system. It's a mixture of macroscopic organisms. You have moles and gophers and earthworms and spiders and beetles and all of those types of things that we're able to see. And then you have all of the microorganisms that are in the soil. You have soil fungi, and you can see sometimes the representative soil fungi on the surface in mushrooms. But those other organisms that you have can be microscopic, so it's very hard to see them, and it's very hard to think that they're present in as high of numbers as they are. It's said that in a handful of healthy soil, there are more organisms than the number of people that ever lived on planet Earth. So we have billions upon billions of microorganisms in that soil environment that are interacting with the environment that they're living in, but they're also acting in such a way that they interact with each other in a variety of ways that's going to allow for some of these processes to occur. And that's also very important because a lot of times a particular type of organism can't really do its full activity alone. It relies upon other organisms to interact with it. The biology of the soil is the organic or living component of the soil. That heart, the soil is the heart of the system. What keeps that heart beating is the activity of these organisms. Again, like I said, there are billions of them, and they represent millions of different species. The total weight of the amount of organisms that are in the soil, in a cultivated soil, you can get it down to about 5,000 pounds per acre. In a very healthy soil, you can have 4 times that amount. And it's difficult to be able to, I think one of the reasons why we don't always look at the soil biology in our systems is oftentimes you can come up with standardized tests that it's fairly easy to look at some of the chemical interactions and some of the physical interactions. But it's not so easy to look at some of the biological interactions because these organisms are so small. You can have a very different community and very different activity that's occurring at one spot and, different activity that's occurring at another spot. And that's going to vary a lot depending on the amount of water or nutrients or physical properties of the soil. So you're going to have these microspaces in the soil environment that may house one type of community. It may be, even though a lot of the soil surrounding it is very dry, there's a microenvironment that's created that is fairly wet. And so it houses a different community than some of the other environments that are in the soil. So a lot of times when I'm doing soil sampling, one of the problems that I run into from a research standpoint is the fact that I'll take samples in a 30 by 60 foot plot, which is about the smallest plot that we set up. So I'll be taking samples from that plot, and I'll take the samples across the plot. And what ends up happening is in one sample, I'll take about 15 samples across the plot, and in sample number 1, I may hit upon a large community of particular organism that I'm looking for. And then in sample number 2 or sample number 3, there isn't that same large community. So it's very hard to interpret exactly what's happening when you can't get a very good count on the number of organisms that are in that whole plot, because there's so much variation between samples. And we're continuing to work from a research standpoint on finding better ways to sample and better ways to analyze the sample so that we can understand that biology even more. These organisms, as I said, there's a lot of interconnection between them because they can't do everything by themselves. And many of you, possibly most of you, have seen this graphic on the soil food web. And the main thing I want to point out with this graphic is that the first trophic level, that first food source, is from photosynthetic activity. It's from either the residue from the plants or from the exudates from the plant roots. And so in order to be able to feed that whole entire food web, you need to have plants that are growing, or you need to have a lot of residue in the system. You need to incorporate something that's going to be putting that photosynthetic carbon into the soil as much as possible. Again, kind of to give you a little bit of an idea of what we're looking at here, we're mostly going to be looking at the microscopic organisms, which the largest ones are 1/200th of an inch. The smallest ones are 1/2000th of an inch. So we're looking at things that are very small by comparison, but again play a huge role, one by the numbers of them that are out there, and also by the activity that those organisms do. So now to kind of take a look at those individual groups of organisms. The bacteria can number in the billions in the soil, and there's only about, there's only about 11,000 different species that may be in a gram of cultivated soil. When you're looking at a undisturbed native type soil or relatively high plant production type of soil, you can be looking at over 500,000 species of bacteria that may exist in there. So there's a lot that happens above ground that impacts the diversity of these organisms that are below ground. The bacteria they primarily feed on organic matter, and it's the organic matter that is easy to break down. So it's some of the more labile organic matter. As they're consuming that organic matter, they're also converting a lot of that organic matter into that humus fraction. So you have some that the organisms are going to take up, but just, again, like all organisms, they take in food and they give off waste. And you're also, in this type of a system, you're both predator and prey at the same time. So we're going to talk about a lot of the organisms that are actually going to feed off of these other organisms, and that's what's going to allow for the cycling of nitrogen and the cycling of carbon and phosphorus and those types of activities in that soil environment. So you have that nutrient cycling occurring. The bacteria can do some things in the soil where they can help to decompose pesticides. They can help to decompose some other chemicals that are in the soil and break that down. And again, they're very important in the nitrogen cycle. One of their important roles in the nitrogen cycle is they act as a food source for larger organisms. And by being consumed by larger organisms, the nitrogen that's in their body is released into the soil environment. You also have a lot of asymbiotic and symbiotic bacteria, such as rhizobium would be symbiotic bacteria that are associated with legumes, that actually can take atmospheric nitrogen and convert it to a plant-available form of nitrogen. So again, they're very important in providing the amount of nitrogen. And that amount of nitrogen is now become pretty close to the amount of nitrogen that we fix synthetically. And we're going to talk about what's been happening with nitrogen use efficiency and the need to keep producing more synthetic nitrogen to feed our systems and how that impacts grain production. Now, the fungi come in two main types. They come in the saprophytic type that feed on the organic matter that is more difficult to break down. They're actually more of the residue decomposers. And that is an extremely important role in our systems, because otherwise we basically would be buried in residue if we didn't have these saprophytes. But the organisms that I spend a lot of time looking at and that I see that play a really important role in our agricultural systems when you look at nutrient cycling are the mutualistic fungi. Primarily they're trading phosphorus for photosynthetic carbon, but they are able to trade a number of different nutrients. They can also act as assistants in some of the other biological cycles that are going on. They can connect one plant to another plant, which can be very important for nutrient flow and carbon flow in the system. They also are very important to the physical structure of the soil, to creating a stable soil environment. And one of the ways they do this is by formation of soil aggregates. The fungal hyphae will act like a net to accumulate soil minerals and organic matter and other organisms to form these balls or pellets in the soil that are the soil aggregates. And the soil aggregates, as we'll talk about a little bit later as well, have a very important role in what's happening in the soil and how that impacts plant growth, as well as the various ecosystem services that the soil provides. But these organisms are, their bodies are primarily these fine threads or strands of fungal hyphae. And those threads are very fragile. They're very easily harmed by plowing or disturbance to the soil environment. And for the mutualistic fungi, they need to maintain that connection with a living plant because they aren't decomposers like the saprophytic fungi, so they can't find a new food source. They have to be connected to a living plant in order to be able to get their food and grow and survive. The mutualistic fungi, the primary group of mutualistic fungi are the arbuscular mycorrhizal fungi. And literally that means arbor, like Arbor Day, is tree-shaped. So they're tree-shaped, and mycorrhizal means myco is for fungus and rhiza is for root. So they're tree-shaped fungal root fungi. And what that means is they are very good. If you look at the picture, you're able to see how the fungal hyphae goes out into the soil beyond that depletion zone that the plant will have. The plants are very good at taking up the nutrients that are right next to the roots or can come in contact with the root hairs. And they deplete the nutrients that are there very readily. But then they need a mechanism to grow out further into the soil. And this may only be 1 to 2 inches out into the soil. It doesn't seem like a lot, but it actually can make a very huge difference on the amount of nutrients and the amount of soil that the plant can access. Again, the fungal hyphae, when they're growing out into the soil, they'll sort of branch out into tree-shaped like structures. And what that allows is it allows them to have a greater surface area to volume ratio. And basically what that means is the fungal hyphae can contact more soil for a smaller with a smaller strand. So there's less carbon that's needed to form the small strands than there are the large strands. And so there's greater volume that's in the large strands. And that allows, excuse me, I'm in my office. We have this system. Sorry about that. So it allows them to be able to acquire a lot more nutrients. These fungi are generalists. They will colonize a number of different hosts. They actually penetrate into the root cell through the root cell wall, and then they'll form these other branch structures. And that allows them to maximize the contact they have with the plasma membrane in the cell, and they can very easily then transfer the nutrients via that contact. So both of these mechanisms are allowing them to maximize their activity for a minimum amount of carbon use. And this is why they're very important to the plants that are growing in the system. If you don't have a lot of these fungi in the soil environment, what we've seen is oftentimes that can stress plants, or it can require the plants to need more synthetic nutrients, because this is the primary mechanism that the plants evolved with over time to acquire the nutrients from the soil. If we take that away, we have to replace it with something. And that's one of the things that's happened in our agronomic systems and the way we manage them. By, again, doing a lot of tillage in our systems, by having non-complex rotations. So you may have corn soybeans, two-crop rotation, or continuous corn, which is only a one-crop rotation. So you're not giving them a lot of different kinds of food. That's what a rotation does, is it provides some different kinds of food to feed these organisms. Again, because they're connected directly to a living plant, if there isn't a plant growing, whenever there isn't a plant growing, you don't have these mycorrhizal fungi growing. So if you have fallow in your rotation system, which is common the further west that you go, you're not going to have a lot of these fungi. And again, what they do is around that rhizosphere, they're very good at they engineer their own environment that assists them in maximizing the amount of nutrients that they can get. They've helped to form those soil aggregates, and in those soil aggregates, they attract different bacteria, and those bacteria will do some of the activity to help to release phosphorus from the soil minerals. They'll do activity where they're fixing nitrogen, and that is providing those nutrients that the fungi can then take up and deliver back to the plant. This is a pay-as-you-go type system. The plant will not give carbon to the fungus if the fungus does not give nutrients to the plant. And again, in our modern agricultural systems, what we've done is we've given the plant a lot of nutrients very early in the growing season, which puts a lot of nutrients in that rhizosphere environment. So the plants don't need to have the relationship with the fungi at this time. And so they'll basically not give carbon to the fungi, and eventually the fungi will start to die off, and they'll kind of give up on being able to get carbon from the plant. And that's what's reducing the numbers of these fungi in the system, in addition with the levels of disturbance and the amount of plant growth that we have. So all of these factors that we're doing through above ground management are impacting how much of this fungal growth we can actually get and how efficient our systems are actually being. This is some microscope pictures so you can get a real visual representation of what's happening. The top picture, the top left-hand picture, shows the fungal hyphae extending out of corn roots. And you can see all of those really fine threads that are out there. And it basically can, if you looked at the amount of carbon that was in the roots and the amount of carbon that's in that fungal hyphae, you can double or triple the amount of access that you have to the soil, but you're only doing that at maybe a half to a quarter of the amount of carbon. So again, it's a very efficient way of working in the system. And the right-hand picture, or the right-hand lower picture, shows the arbuscules that are inside the roots. And as you get more of that tree-shaped or bushy structure, the more you have in contact, more hyphae you have in contact with the plasma membrane of the root cell, that allows you to have this very efficient type of exchange. So again, these organisms evolved with the first land plants. For plants to be able to grow on land and get the water they need and nutrients they needed to be associated with these fungi. There's strong evidence of this in the fossil record. So we're able to see that these plants didn't really utilize their roots. In fact, the first land plants utilized their roots primarily for a anchoring structure, so that they could start to grow up as opposed and get into larger sizes, as opposed to algae that are in aquatic systems that they originally evolved from, which are mostly unicellular. So they're now able to become multicellular organisms and grow up because they're anchored in a spot. That anchored in a spot makes it harder for you to get nutrients. And so they had to form this relationship in order to be able to get the nutrients they needed from the soil. The next organisms we're going to kind of take a look at are the nematodes. And I wanted to bring up the nematodes because nematodes kind of generally get a bad rap. They're mostly considered bad, they're pathogenic, they're horrible to have in your system. You have to put in pesticides and exterminate them as much as possible from the system. Most of the nematode species are actually non-pathogenic. They're organisms that actually primarily feed on fungi and bacteria. And that can be very important because that means they're very good at helping to control the populations of some organisms. The idea of the diversity of these organisms, like I said, you can be both predator and prey at the same time. And by being prey, it means that there's one organism that helps to keep your population in check. The reason that we have a lot of diseases in our modern agricultural systems is because we're growing primarily one type of plant that is supporting the life of very particular types of organisms in the soil. And those numbers of organisms, because they're the only ones getting fed, their population grows out of control. But if you maintain the diversity of the system, those different organisms are going to make sure that one population doesn't grow out of control. So it's very important to have these organisms, and by consuming the bacteria, they can consume the bacteria in very large numbers. And so it's very easy for them to release some nitrogen from the system, because the nitrogen, again, that's locked up in the body of the bacteria is consumed by the nematodes, and some of that nitrogen comes out of the nematode as waste. And then that nitrogen, when it comes out of the nematode, is in a plant available form because it's been broken down. So it's in a inorganic form instead of the organic form that it was in when it was a part of the body. And when it's in that inorganic form, it now can be taken up by the plant. So it's a way of cycling nitrogen through the system. It goes from an inorganic form into the bodies of the bacteria, and then the bacteria are consumed, and it's released from the bacteria into an inorganic form. And that can go back up to the plant and be converted into an organic form again and keep on going through the cycle. Unlike the bacteria, where you have billions of them in a gram of soil, for the nematodes, you may have only 10 to 20 individuals. But again, their size is much larger, so they're able to, those 10 to 20 individuals can represent about 10% of the microbial biomass, whereas bacteria will represent about 30% of the microbial biomass. So in a lot of the cases with the organisms below ground, size and numbers matter. And you want to make sure that if you're small in size, you have a very large number, so that you can actually impact the entire system. The picture that's at the bottom is one that I like to show because it's actually a fungus that is trapping a nematode. The fungal hyphae, those threads that are the body of the fungi, it forms a loop or a lasso, and then it will give off some compounds that will attract the nematode. That basically is telling the nematode there's food there, and the nematode will come toward it, and when it wiggles its way into that lasso, the fungi will actually cause cytoplasm to get into the cells that are making up that lasso, and those cells will swell, and then it traps the nematode there. And then the fungi will release enzymes that decompose that nematode. So they're actually, again, this is a way of the predator and prey relationships. Not only do you have the nematodes that consume the fungi and the bacteria to keep their numbers in check, but you also have fungi that can consume nematodes to keep their numbers in check. So again, you have less pathogenic type of relationships in a healthy soil than you do in a non-healthy soil. And it's because of all of this activity of these organisms. They're helping to keep populations under control. The last microorganism, I'm not going to discuss some of the meso and the macroorganisms very much in the system, because I want to focus in on some of these very small organisms that we don't see very much and we don't pay a lot of attention to. So the last ones I'm going to focus on are the protozoa, and they include a wide diversity of types of organisms. Many of them that you've probably seen or heard about. You have the paramecium, you have amoebas that basically move by flowing their cellular contents from one part of their body to the other part of their body so that they can move through small spaces and actually move in the soil environment. Most of the ciliates and the flagellates actually have hairs that help them to move. The organism that's in the upper right is sort of a cross between the two. It has cilia, and you can kind of see they're fuzzy on the front part of the organism where its mouth parts are. And that cilia, what that does is instead of helping the organism to move, it actually causes bacteria to flow towards the mouth of this particular protozoa. So it makes sure that it gets its food source flowing to it. But it actually moves more like an amoeba. It moves by moving its cellular contents, or it's called pseudopodia. They stick out part of their body like a leg or an arm and move that, and that allows them to move forward. It's an anchor that they can move the rest of the body too. And again, like the nematodes, the protozoa are larger in size, so there's fewer in number in a gram of soil, and they consume the bacteria. So again, they're an important part of the nitrogen cycle because they're eating bacteria and pooping out nitrogen in a plant-available form, in an inorganic form, when they break down the bodies of those bacteria. Now, oh, sorry, I have one more group. I apologize. One more group, the microarthropods, and these are the microscopic insects, and they are the decomposers and the shredders. They're very good at breaking large pieces of organic matter down into smaller pieces that then the bacteria can consume. They are, however, in an agricultural system, they are very easily harmed by tillage practices and pesticides. The insecticides that we utilize in our agronomic systems to control large macroscopic insects also affect these microarthropods, these microscopic insects, which means that every time you spray an insecticide, not only are you getting rid of all of the larger macroscopic insects, whether they are good or bad, you're also getting rid of the microarthropods, whether they are good or bad. And that can affect the amount of food that the bacteria can have because there's nobody to shred that organic matter into the small pieces the bacteria can eat. So organic matter decomposition relies more on the activity of saprophytic fungi than it does on this combination of microarthropods and bacteria. So now you're limiting those various populations as well. And this kind of gives you a relative size of things. So we've got a sand particle, and then you've got one of these microarthropods, these microscopic mites. That's the biggest thing on the picture. And then you can hardly see the nematode that's there. It's a microscopic worm. From a scale standpoint, if you were looking at bacteria, you basically would see instead of a mite, it would be a nematode that would be that size, and then the bacteria would be something you could barely see on the screen. So again, there's a huge variation in size in these microscopic organisms, but they all play some different roles in helping that system work. There's also earthworms that are in the system, and this is the only macroscopic organism that I'm going to talk about. And the reason I'm bringing up earthworms is they form, and you can see it in the lower right-hand corner, they form these tunnels in the soil, and they leave behind on the surface of the soil casts that contain a huge amount of nitrogen and phosphorus and organic matter, and that helps to feed some of the other organisms. In fact, along that tunnel that is growing through the soil, you'll have bacteria that will just line the surface of that tunnel. Because as the earthworm grows through the soil, it leaves behind, on the surface of the body, it has some lubricating polysaccharides. And it leaves behind those polysaccharides, which then becomes food source for bacteria and fungi. So it's a way of the fungi and the bacteria getting deeper into the soil and having a continuous amount of food source, because they can't travel very far given their limited size. So kind of the bottom line with what you want to do, the first thing that you really want to focus in on for the system is to replace iron tillage with biological tillage, with the activity of earthworms. Replace it with the activity of plant roots that you have some large taproots that can help break up layers of compaction. The fine roots that will come from grass plants that will spread out and further into the soil and create smaller channels, but they're still creating tunnels and reducing compaction in soil environment. And I say it's the primary thing to do, even though the greatest need that a plant has is for food, because a lot of times if you have a lot of compaction and you have in an iron-tilled system, in a conventionally tilled system, you'll have layers of compaction that are right under that plow layer. And that means that you can't get the plant roots and you can't get those organisms that grow with the plant roots very far into the soil environment. And so you aren't really being able to support a lot of good diversity of organism growth. They may have food because you have a plant growing, but their habitat is so disturbed and so broken up that it's hard for those organisms to even try and survive and to be able to get to many of those food sources. So again, what you want to do is you want to build the amount of organisms that you have in the soil. And I want you to do cultivation, but I don't want you to do cultivation with a piece of metal equipment. What you do cultivation with is you cultivate those organisms. So they will do that cultivation of the soil environment themselves. And again, what they need primarily is food. You need to have a diverse crop rotation. So you're feeding a different type of food source. This is basically giving the soil environment proteins and fruits and vegetables and breads. All of those things that the different organisms need to grow, you're providing that into that environment when you have a more diverse crop rotation. And if you're in a system, you're working with producers that are in a system that, because of equipment and because of marketing needs, it sort of fits them better to have a shorter rotation. There are alternatives to improving that crop rotation by adding cover crops or switching it up a little bit where maybe you'll grow a cover crop for part of the year or you'll grow a hay crop for part of the year so that you've got something different that's in that system that helps provide that diversity. you want to make sure that that food source is consistent. The last thing you need are these organisms starving. And Jim Horman brought this up. He was talking about the fact that, you know, these organisms, even here in North Dakota, which many people consider the end of the earth and a frozen wonderland, even here in North Dakota, we can have microbial activity happening almost every day of the year. And I say that because we've done studies where we've looked at gas emission. We've looked at CO2 emission and nitrous oxide emission, gases that are produced by organisms that are growing in the soil environment, and we can measure those almost every day of the year. So we're able to see the fact that organisms are growing, and part of that is those microorganisms are creating those microsites. They're creating that little environment that they need to be able to continue growing. They'll create those stable aggregates that are part of their habitat. They engineer their own homes in order to keep themselves in an environment where they can maximize the amount of food that's available to them. They can protect themselves. The bacteria that are in these aggregates are protected from the nematodes and the microarthropods and the protozoa, so they're not consumed at the same rate. And they're in an aggregate where there's organic matter available to them. It's locked inside that aggregate that they can consume. And that aggregate, again, it's a protected environment where a lot of the changes in temperature and the changes in moisture, that's kind of the last places they'll get to is inside that aggregate. So you can have more stable temperatures and more stable water conditions, moisture conditions, to maintain that activity for a longer period of time. And what we do with a lot of our systems when we don't include cover crops or perennials, something that gives continuous cover, what happens is we're basically feeding the organisms for, if you're growing a short season grass like wheat, for about 2 months of the year, and expecting those organisms to survive until the next year when you're planting. So we're feeding billions of creatures on about 2 months of carbon and expecting them to survive for 10 months. And it's a system that over time doesn't work because you're continuing to deplete the number of organisms that you have in your soil. And again, if you cultivate these organisms, you're going to increase the amount of soil organic matter you have. You're going to improve the fertility of the system because you're going to have better nutrient cycling through all of these different types of organisms. And it, in the long run, will increase the profitability for the producer because there's less synthetic nutrients they need to add, less pesticides they need to add, less maintenance and cost in creating a non-compacted growing environment for the plant roots to be able to grow so you can get higher yields. And you're also creating an environment where you have less topsoil loss to erosion, whether it's wind or water erosion. So you're creating a system that is automatically going to be more profitable to the producer. In the beginning of creating a lot of these systems and making these systems changes, you may end up seeing yield declines. It's very common as the system's transitioning from a system that wasn't very biologically active to a system that's very biologically active. But at the same time, you're reducing costs. And so a lot of times when you're, I guess, trying to sell these management changes to producers, when I'm talking to producers, a lot of times it's not just about what you make at the end of the year when you sell your grain, but it's about what your overall expenses were, your overall net profit was. And that's what's going to be important in making sure that people kind of utilize these systems because you can reduce some of these costs, reduce the amount of passes that you have over the field and reduce the amount of diesel fuel you use. And like I said, you reduce the cost of pesticides and the cost of some of the synthetic nutrients. And, you know, from an environmental standpoint, you also get long-term sustainability of the system. the way that we utilize a lot of our agricultural systems right now, we do different mechanisms in order to try and make them sustainable. We do terracing. We've done sustainable tillage. We try and grow more crops. We try and increase plant population. All of these things that we're trying to do to help create a more sustainable system. But at the same time, a number of those things that we're trying to do are actually not contributing to long-term sustainability of the system. It's a very short-term impact. And what we need to do is introduce all of these tools to create a system that's biologically active and going to be able to provide these benefits. And this is just a kind of a long list. I'm not going to go through it because we're kind of going to touch on a lot of this stuff and have touched on a lot of this stuff. But these are the things, as you can see, there's a huge amount of things that just putting carbon into that rhizosphere, increasing the amount of root exudates that you have, can have on the health of the entire system and sustainability of the system. And when you do these things where you're changing the tillage type and you're changing the plant diversity, adding these various components, and you don't have to add them all. Just as you're adding some of these various components, what you see is that the math doesn't work anymore. 2 plus 2 does not equal 4. You can have 2 plus 2 equaling 16 because the effects are not adding, they're multiplying each other. And so you want to be able to create these management systems that are actually going to give more than they're going to take away. And that's what, working with the above ground management and reducing the amount of tillage, adding some crop diversity, adding cover crops, adding manure and compost, working with the timing in which you make these changes, adding grazing as a way of managing some of the forage that you have, some of the crop residue that you have, making sure you maintain some crop residue in the environment. All of these different things are going to have huge benefits to the system. When you do tillage, I talked about this with the fungal hyphae, and this sort of illustrates what happens. So you do tillage, and it breaks up the fungal hyphae into thin strands, and it exposes that humus organic matter to the surface. And when that humus organic matter is on the surface, it's actually exposed to the decomposing organisms. And so it basically can get eaten up very fast because the decomposing organisms, what they do is they eat high carbon, low nitrogen material, and they eat it fast. And the reason they do that is because oftentimes the protein, or what contains the most nitrogen, is taken off of the field as the crop that you're growing, as the grain that you're growing. And the residue that's left behind is very high in carbon and low in nitrogen. And that's very similar to that buried organic matter, that humic organic matter in molecular structure. And so those decomposers that need to eat it fast so that there's not residue that is increasing on the top of the soil, so that you're getting some residue decomposition, they eat it very fast. And they'll eat the organic matter as well. And then that just ends up getting burned off into the atmosphere as CO2. And we've seen evidence of that as we've looked at the historic loss of soil carbon. So as we're looking at when we started applying tillage to the soil system, we rapidly reduced the amount of carbon that was in the soil. And we're trying to change some of that. You can see sort of starting in around the 1970s when we had the oil crisis, and then we had the Clean Air Act put in position and the Clean Water Act put into position. Those changed some of the ways that we managed, but that actually hasn't had a dramatic increase in the amount of carbon that we have in the system. So we want to increase that amount of soil carbon because we could see all the benefits that come with water holding capacity and food for microbes and all of those different things that come with having more organic matter in your system. So we want to continue with this idea of reducing tillage, but we also want to find some other mechanisms that may allow us to increase the amount of organic matter we have in our soils at an even faster rate. And one of those is by adding crop residue. And, there's a lot of question as to if residue is treasure or trash, if it should just be taken off and maybe utilized as a biomass fuel source. And, there's issues with it for seeding. There's issues with it for carrying disease, all of these different types of things that we think that we don't want to have a lot of crop residue. And in corn cropping systems, they actually add a lot of nitrogen to help to decompose that residue at a faster rate. But why would you want to do that? Because the residue is protection for the soil. It is providing some nutrients to the growing plant as some of that residue is decomposed. It's providing more organic matter and much more food to the organisms that are below ground that allow for those organisms to grow and provide all of those benefits we talked about before. So maintaining residue can be one way to couple with tillage that's going to help to improve the amount of carbon that we have in our system. Plant diversity is also going to help with the amount of carbon that we have in our system. And you'd think that if you added a lot of cereals, which have a high carbon to nitrogen ratio, meaning for every atom of nitrogen, there are 80 to 100 atoms of carbon. And you would think that that would be good, because the more carbon you have, the more organic matter you have, right? Well, that's not really the case, because again, when you have that high carbon to nitrogen ratio, the things that are decomposing that are very fast-growing saprophytes. They let most of that carbon go back up to the atmosphere as CO2, and the nitrogen is consumed in that decomposition of that carbon. So you don't end up building a whole lot in that system. But if you add things like legumes, they're going to increase the amount of nitrogen that you have in the soil, and they're going to improve that system so we can get better, we can get closer to what the average is for soil organic matter itself, closer to that 10 to 1 or 12 to 1 type of organic matter, that we're better able to build up that instead of consuming all of the nutrients in trying to decompose that. Now, cover crops are a thing that we're discussing more and more often as possible mechanisms to improve the system. And one of the things that cover crops is doing is it's continuing the amount of living plants that are growing in the environment. And when you continue the amount of living plants that are growing in the environment, you're getting more carbon exuded out of the roots, you're building up that organic matter in your system, you're providing more for the mycorrhizal fungi, for the mutualistic fungi to be able to grow because they're directly connected to the plant. And from the mutualistic fungi, they're giving off carbon to bacteria. They're providing a food source for some of the larger organisms in the soil, the nematodes and the protozoa. So it's a very good way of being able to get more carbon cycling through and feeding these different organisms. And that's going to help overall in building up the amount of carbon that you have. I want to kind of point out these pictures. The top one was actually from my dad's farm in southwestern Minnesota. He does sustainable tillage. He's not into no-till. And, corn, soybeans, he's now converted mostly to corn in his system. Corn and wheat, actually, he's kind of changed up in the last 2 years. But what he's done is last fall, he came up here to North Dakota, and he heard a number of producers talking about the benefits of cover crops. So he put in a cover crop into his system. And then this spring, he burned off the cover crop that regrew, even in Minnesota again, where it gets fairly cold over the winter. He did have cover crop that he planted last fall, and that regrew. He planted it about August 20th, and that regrew in the spring. And he had clover and radishes and turnips in the system in his cover crop mix. And so he got a lot of clover this spring and a little bit of turnips growing as well. And then he planted it, he burned it down, and he planted it to corn. And this is on June 19th. It's about not quite a month after he planted the corn, but the corn is coming up through the stubble that he had left behind. It was a very cold spring, so you wouldn't expect it to be very high, but it still shows you that you can add some of these things to your system, and it's going to be able to continue what you want to have happen with your cash crops in your system. There are mechanisms to do it. There are plant choices that you can make. There's timing choices that you can make. There's, cash crop choices. There's also cover crop choices that you can make that are going to help make this work. The bottom picture is actually a very interesting producer from Emporia, Kansas, and this is his corn crop, which was planted in June after he harvested a winter wheat crop. And then with the corn crop, he put in a cool season cover crop mixture. So it would grow as an understory crop with that corn crop, and some of those plants are going to survive in the shade under that corn crop and be able to grow into the fall after he harvests the corn. So he's maintaining that living cover as long as he can. And that's, again, one of the biggest keys about improving the system. Again, you have lots of different cover crop choices in the plants that you can choose: warm season and cool season, legumes and grasses and oil seeds, just a tremendous amount of choices that you can make in order to tailor that cover crop for what it is that you need to tailor it for. This is a picture you probably have seen before. This was up here in North Dakota in 2006. We had a total, where this field was at, a total of 7 inches of soil moisture. And where they planted one species of cover crop, the crop died. It was a very dry, obviously, and it was a pretty hot year. Where they had the cattail, it included both turnips and oilseed radish. They had a mixture of that with a number of different crops, and that crop thrived. These pictures were all taken on the same day. Now, one of the reasons that can work is that you can get these things where, again, the effects are multiplying instead of just adding together. So if you add no-till and crop residue, and then you can also add a cover crop, so you've got living plants that are growing, you're definitely going to have a lot less weed pressure, because the seeds are not going to be able to germinate very well because they're not going to get a lot of sunlight. There's poor seed-to-soil contact, and the seeds with the no-till system are left unburied, as opposed to when you till, you basically are burying weed seeds. So you're able to get this system where you're getting an increase in weed control without having to use any type of pesticides to do that. Now, this is where I'm talking about fertilizer use, especially nitrogen, which are the blue diamonds. There's been a huge increase in the amount of nitrogen that we're using in the United States in the last about 50 years. It's not been a whole lot of change in the amount of phosphorus and not much change in the amount of potash. But focusing really on the nitrogen in the system, you can see this was done in Illinois at the Morrow plots, and they were measuring potentially mineralizable nitrogen, so the amount of nitrogen that can become plant available. And what they found is over time they had a continuous corn treatment. They had corn and oats. The oats were replaced with soybeans in '67, and they had a corn oat hay type of system. And they applied new nitrogen synthetically as was applicable to the particular crop. And most of that had been the same since 1967, some of it since 1955. But what they found is that between 1955 and 2005, they would have a drop, obviously, in the amount of potentially mineralizable nitrogen. So the amount of nitrogen that you could get that would feed your plant, even though the fertilizer rate was the same. There was this drop in the amount of nitrogen that was available. So something's happening in our systems that, again, is making that nitrogen use efficiency decline. And this was done up in Canada, and what they found is that the longer they had no-till in the system, the more their production didn't change, even if they added more nitrogen. So if you were in a conventional system or transitioning out of a conventional system, you needed a lot more nitrogen to try and get very similar yields, instead of being able to maintain yield with very low amounts of nitrogen and utilizing that nitrogen that's in the soil system. We're seeing that this graph is again showing on the left-hand side the cereal yield over time, and that seems to be following fairly closely the amount of nitrogen that we've applied. It's the same shape of graph. But when you actually look at nitrogen use efficiency, so what you are really getting back over time in yield compared to the amount of nitrogen that you're adding, you're actually seeing a declining curve. So that's looking at, it's not looking at grain yield increasing because of changes we've made in management, or nitrogen use increasing because we're increasing the amount of nitrogen, but how those, when you put those graphs together, how they actually look together. And even though you'd think that they, look fairly the same, that would be an inclining curve, it's actually a declining curve. So we're seeing that when there is more fertility added to the system, there's not necessarily more yield. And a lot of that has to do with the timing and the availability. And when you work with the organisms in the soil, they're actually going to change the amount of nitrogen that's available and when it's available. You'll get these things where you get interplant transfer. You have basically a corn plant and a legume plant growing side by side. And in the rooting environment, you have fungal hyphae that's connecting those two plants together, and you have rhizobium bacteria that are fixing nitrogen. And that rhizobium bacteria, to fix nitrogen, it takes a lot of energy, just like it takes to make synthetic nitrogen. And that energy comes from the cellular powerhouse, which is powered by the amount of ATP. You basically take this molecule called ATP and you break off a phosphorus, and that releases electrons to help fire the energy for the different processes in the cell to work. And when you do this, you actually need an influx of phosphorus to convert that ATP, that ADP, it goes from ATP, which is adenosine triphosphate, to ADP, which is adenosine diphosphate. And to put it back to ATP, you need an influx of phosphorus. When the mycorrhizal fungi are associated, in particular with a grass plant, they have better mechanisms, even better than their natural mechanisms to acquire phosphorus. They work with bacteria that are actually in the fungal hyphae that colonize the surface of the fungal hyphae, and those bacteria solubilize phosphorus from minerals. And so it's a way for them to get more phosphorus. Having them associated with a grass plant stimulates the amount of activity that occurs and the amount of phosphorus that's acquired. So the fungus will take some of the phosphorus that it acquired for the grass, and it will transfer it to the legume. So the legume can transfer it to the rhizobium. So the rhizobium can fix more nitrogen, and then the fungus will take that nitrogen and transfer it back to the grass plant. And we're really seeing this effect happen when we have these cover crop cocktails, crops that are growing at the same time. So again, we're getting improvements in the cycling of nitrogen when we have cover crops in the system. So we're now going to try and improve that nitrogen efficiency, because now that we have improvements in the amount of nitrogen that can be fixed and the way the nitrogen can be transferred between the plants, it allows us to use a lot less fertilizer, because it's providing that nitrogen fixation, is providing a continuous nitrogen source to both plants in the system. and then their residue is going to have more nitrogen in it, and you've got a legume that has a higher amount of nitrogen compared to the amount of carbon that it has. So it's able to actually get more nitrogen in the residue, it's able to get more nitrogen in the roots, to be able to feed these organisms in the soil, but also to be able to create nitrogen for the subsequent plants to be able to use, and we have to use less fertilizer to do it, and that's what's going to improve our nitrogen use efficiency. The cover crops, again, can augment the amount of soil cover that you have, and even when you just have residue on the soil, you're not able to cover as much of the soil surface as you would by the shading of leaves of different sizes. So when you have a cover crop that has a mixture of different plants that have a number of different leaf sizes and leaf heights, you're able to better cover that soil surface, and you're able to, this was again done in North Dakota on those fields that the previous picture with the dead radishes and the dead turnips and the living cover crop mixture, you're seeing about a 20-degree temperature difference between having that cover and having just some residue that made it through the winter and spring on that soil. And that makes a huge difference in the amount of microbes that you get. Optimal microbial growth occurs at about 80 to 100 degrees. It starts slowing when you start going above 100, and as you get from about 100 to 115 to about 130, it basically stops. It kills off all of the microbes. So at the previous slide, at those temperatures, what you're getting is you're getting optimal microbial growth in the field with the cover crop plant, and you're getting a slowdown and almost a death of microbial growth when you've got the soil with just a little bit of residue cover. If you want more information about cover crop species and their growth cycle and those types of things, we have a tool. It looks like a periodic table. You actually click on the different plants you're looking for, and it will open a window that will give some more information about those plants. So you can choose and tailor your cover crops to what it is that you need to have them for. You can make sure that the system is provided with everything that it needs to be able to function. Things that are going to be able to grow at the different times that you want them to grow, things that are able to use more water or use less water depending on your moisture conditions. All of those things you can pick out, and you can tailor this crop to be able to function with. I'm going to try and talk very quickly. I know we're kind of going over time, and I apologize for that. I want to talk quickly about soil aggregates, these pellets that are in the soil. In the lower right-hand picture, I want to point this out because a lot of people are, my soil is too sandy. I can't grow stuff here. I can hardly grow my cash crop. How can you expect me to grow my cover crop? I can't get all of these benefits because I can't get organic matter in my soil. All of these different things that they're talking about. This lower right-hand picture is actually from a pot culture system that has been growing for 3 months. And when I'm doing experiments in the greenhouse, a lot of times I use a 1-to-1 sand-soil mix. And so I added a lot of sand, and you can see that it's fairly sandy in there. I added a lot of sand, and after 3 months, I'm still getting this level of aggregation. The picture on the lower left is actually a millet root that was planted as part of a cover crop mixture. This sample was taken from a plant that was planted on July 7th. I collected these roots on August 31st, and the previous crop before the cover crop was put in was a forage pea. But this is on the particular cover crop root, and you get all of those aggregates that are formed in that rhizosphere environment right around the root. You can't even really see the root. It's hidden by all of these aggregates. So again, in a very short period of time, in very sandy soil, the picture on the left was actually, it's a sandy loam soil that it was growing on. So, in these soil environments, you can still get a high level of soil aggregation. These soil aggregates, again, I said were formed by the net of the fungal hyphae. The fungal hyphae you can't see in this picture. It's clear hyphae, but you can see the effect of it because it's holding those clumps together. Those clumps right now are primarily just, I separated them from the soil by wet sieving, so they're primarily just plant debris and fungal hyphae and a little bit of clay minerals and possibly a little bit of silt minerals, but there's not very much sand in here. And you'll see how the fungal hyphae, I did wet sieving, so I broke this up, and then I washed it off of the sieve into a glass plate, and I let it sit for a couple of hours, and it started forming these nets that started to bind all of this material together. So this is sort of a pre-aggregate formation. When you get aggregation, you reduce the amount of compaction in your system. You're going to get those pellets come in different shapes and sizes, and so they don't fit together exactly. And so they create space in between them, and that space in between them is the porosity, the amount of porosity that you have in soil. And when you have greater porosity, you're going to have greater levels of microbial activity and microbial growth, because that porosity helps moisture get into the soil. It also helps air to get in the soil, and it helps CO2 to escape the soil. Most of the organisms that are in the soil conduct respiration the same way we do. So they take in oxygen and they give off CO2. But they're again trapped in this environment, and the amount of compaction that you have when you reduce the level of porosity, because you're in a compacted environment, the less those gases can exchange. So the less microbial growth you can get in that environment. Water management is an important part of having these aggregates, because you're creating that porosity where you can get water to go into the soil much more quickly, and you can keep water stored in the soil for a longer period of time because it'll be stored in that pore space, in that open space. And I like to, this was a study that was done in Missouri, and I like to sort of point out that kind of it was a summary on several studies that were done called the drought myth. And basically what it said was that you could have unfertilized corn that would require 26,000 gallons of water per bushel. But if you fertilize that field, you only need 56 gallons of water per bushel. And what it is that what's happening here is as you have greater fertility in that soil environment, the plant roots, in order to get fertilizer from the soil, give off water to create mass flow for the different fertilizer, for the different nutrients, for nitrogen to flow towards the plant roots, for phosphorus to flow towards the plant roots. They give off water to do that. But if you can circumvent the amount of water that they give off, either by adding a lot of synthetic fertilizer really close to where you plant the seed, so that the plant is able to acquire a lot of that fertilizer and utilize it with very little water give off for the fertilizer to move, or if you can circumvent it via the mycorrhizal fungi, which I said are very efficient at going out from the plant roots, so the plant roots don't have to give off any water. They give a little bit of water to the fungal hyphae, but for the most part, the fungal hyphae acquire that water from the soil. So the fungal hyphae grows out of the roots and then picks up those nutrients and carries them back to the plant. So you don't get a lot of this water loss, and it's going to require less water in your system. As you increase the porosity level of your system, you get 44% greater. As you increase the amount of porosity you have in the system, you can increase the amount of infiltration. And just if you increase the porosity by about half, you can go from 167% to increase in water infiltration for the first inch and 650% increase for the second inch. So it dramatically increases the amount of infiltration you have. The porosity also can reduce the amount of drying that you have, because basically what it forms is as you have those aggregates that are there The water has to move and curve and bend around where the aggregates are, because those pores aren't straight channels from the surface to into the soil. And because of those moves and those bends and the curves, the evaporation off of the soil surface is slowed. It's basically like putting bends in a straw. If you put bends in a straw, it takes more energy to try and suck up the water that's at the bottom, versus if you don't have bends in the straw, it's really easy. You don't have to suck very hard. So what you want to do is you want to have bends in the straw that's connecting the above ground to the below ground, so that more water is going to stay in that environment. Water infiltration rate increases. You've got a conventional system that's on the left-hand side, conventionally tilled, and as you can see, most of the soil that's at the bottom, this was down about 3 inches. This was from a rainfall simulator. You can see most of the soil at the bottom is not wet. But if you go to an undisturbed system, a no-till type system, what you're getting is more water that's making it through. So this is saturated all the way down 3 inches. It also has higher organic matter levels than in the conventional system, as you can see by the color differences. So when you get aggregates in the system, you're going to improve porosity, which is going to reduce compaction, allow for better root growth. It's going to allow for better aeration, better water infiltration, better water holding capacity. And the aggregates are also going to be a lot less susceptible to erosion, because when you have material that's bound together in a pellet, it's larger in size, and it's going to take more energy, stronger rainfall or a stronger windstorm to be able to move that than it is going to be for all of the fine particles that would be individually in the soil. And it's going to improve the nutrient cycling by providing a protective habitat for the bacteria. It's going to protect organic matter, and that's going to be a good source for the bacteria as far as food is concerned. So you're going to be able to have this better environment in the system for better root growth and for better microbial growth. This basically just illustrates some of the management impacts on the amount of aggregates and the stability of those aggregates. In the top 3 pictures, you have one that came from a conventional till spring wheat fallow system. Next one is a no-till spring wheat, winter wheat sunflower system, and the last one is a moderately grazed pasture. These aggregates were separated from the soil by passing them through a series of screens. So they're all 1 to 2 millimeter aggregates. They're all the same size. When you add the same amount of water to these dishes, what you'll find is the WSA, or water stable aggregation, increases as you go from a management system that has less soil disturbance and more plant growth for a longer period of time. And you also increase the amount of TG, which is a total glomalin, which is a polysaccharide type of substance that's produced by mycorrhizal fungi that helps to contribute to the stability of the aggregates. As you change management, you actually impact on a relatively small scale these aggregates, and that's going to have a really strong impact on all of these other activities that you want, reducing compaction, erosion, and the like. So what you really want to do is focus on management and not just getting the job done, not just completing it, but actually looking at the steps that you utilize in your whole entire system. And I believe that the new green revolution is actually going to be a brown revolution. It's going to be looking at the soils and soil environment, figuring out how to utilize resources the most efficiently, getting those resources into where they need to get in with the least amount of loss of water and the least amount of cost of carbon. And this doesn't really mean that you have to work all that much harder. You just have to work smarter. You have to, again, look at your whole entire system. You have to look for what you need for pest management and what's the best, most efficient option for that. Look for increasing the balance in your carbon and nitrogen ratio. So having high carbon, low nitrogen, and low carbon, high nitrogen plants in your system. You want to make sure that you protect your soil, keeping it covered by living plants with different leaf sizes and different placements, or by having residue on that surface of the soil. You want to keep that soil environment protected from temperature increases as well as rainfall and wind. So you want to make sure that that environment stays there. And you, again, really want to use your water resources and your nutrient resources most efficiently. And it doesn't matter where you are, that's what you want to do. Whether you're in a dry environment or a humid environment, a cold environment or a warm environment. All you want to do is improve the efficiency. So what are the steps you can take to do that? And thank you. Again, I apologize for taking so much time. If you want to contact me, that's my contact information at the bottom. And I guess we'll go for some questions.
Yeah. I hope everybody appreciates the presentation you just heard. If you've ever had to make a presentation sitting in a room by yourself and to cover a topic so thoroughly and discuss it for 90 minutes, I'm very impressed. Fortunately, we've been able to capture this, and it's been recorded, and I'll show you in a second where you can go back, because I'm sure a lot of us are going to want to do that. Operator, right now we have opportunity. Let's just take a few minutes. I know we're running over, but hey, we got nothing else to do. Take a few questions and then and see what's anybody's out there. I'm not sure. There might be any. I had a couple here. There's a lot of controversy out, I know, in the Great Lakes area, Chris, about earthworm holes and the movement of, especially dissolved phosphorus. You got a comment on that? Is that, have you done any research or read anything on that? They're actually blaming some of the contamination of the Great Lakes on no-till and water movement down into field tile. Have you got any experience or comment on something like that?
I guess I would say I don't have much direct experience with some of that. As far as the earthworm tunnels go, I would say that although they're creating a tunnel to be able to get water into the soil pretty quickly, and that can move some phosphorus and it can move some nitrogen into that soil, and that can get into your tile drainage system and possibly even get into the groundwater system a little bit more quickly. But what they are doing as far as increasing the amount of carbon that's going to be available to the different microbial species that are going to be able to utilize that nitrogen and phosphorus in a way that's going to keep it out of the river systems and lake systems is going to, in the long run, be more beneficial. There may be, just like anything, there may be a few places in which it's not working quite as well as it should, or the system just hasn't gotten to that point. The other thing, by having the microbes growing along those tunnels and the polysaccharides that are left behind by the earthworms, that can really help contribute to aggregate formation. And when you contribute to aggregate formation, that's another way of locking up some of those nutrients, because they're going to be inside the aggregates, and their aggregates again are going to create those tunnels and those channels that are going to have curves and bends in them, which will slow the rate of flow of the water as well as those nutrients.
Okay, thank you. We had a question come in, and somebody wants to know what the role of higher animals, grazers, in the overall nutrient cycling and in this whole discussion about improving soil quality and the soil biota. How significant is that to work into a system?
It's something that we're learning more and more about. Again, there's been a lot of decoupling of grazing systems from agronomic systems, and as we see those systems come back together, we're actually seeing better nutrient cycling. And there's something that goes on with grazing that I don't fully understand, and I don't think anybody fully understands it, because we try and look at the microbes that are left behind in the manure, or the microbes that are growing in the near and where urine is excreted, and we're not able to figure out exactly what's happening, but there's something that is occurring with, possibly with what types of organisms they are or with some of the activities that they're having in that particular environment that can't be replicated by haying it off or, you know, mowing down that residue. There's something with having that higher animal on the fields and on the system that's going to allow for better management of those nutrients. And especially if you're putting them, the livestock, out to graze the fields or graze pasture systems rather than having them in feedlots, because you're getting a greater distribution of those nutrients that come from the manure and from the urine, as well as the microbial populations that come out of the ruminant with the manure.
Okay, I got two questions, and then we'll close this thing off. You introduced a new term, sustainable tillage. What is that?
Sorry. Sustainable tillage is going away from the moldboard plow. So it's sort of a reduction in tillage. I heard about it as a kid from what my dad was doing when they went to these different things with doing terraces and going away from the number of passes that they were doing. So they were doing fewer passes in the fall and you know, trying to just create the seabed in the spring. It's still doing tillage, but it's sort of modifying it from kind of the more conventional, intensive, multi-pass, deep tilling type of equipment.
Okay. We probably coined the phrase conservation tillage or
conservation tillage, yeah.
Okay. I had a question come in about the use of BT crops and what is the effect that these are having on biological activities. And is there going to be a problem with excessive residue because we've heard that there's a slowing down of the breakdown process using BT crops?
Yeah. I have also seen quite a bit of data that's looking at the slowing down of the residue decomposition of BT crops. I've looked at several different research papers. I haven't conducted research in that area, but I have some colleagues that have. And there are some that have looked at the microbial community and have said there's no difference between BT corn and non-BT corn. And there are some that have said that there is. And a lot of that, I think, does have to do with where you were sampling. Like I said, sometimes you'll hit the spot that is the greatest spot on Earth, and sometimes you'll hit the poorest spot on Earth in the same field under your sampling regime. So I think that we're going to find that a lot of it has to do with that. We're finding out more and more about what's happening in the rhizosphere environment with introducing genetics and introducing some of the pesticides that we use and the impacts that that's having. And that's just, I think I'm going to say more of a stay tuned than we really have any answers, but we're definitely working there.
Okay. One last question. The soil food web analysis that's available out there from, well, I guess it's from the soil food web. You got a comment on that? Is that something that's worth pursuing or?
I do. The soil food web analysis, and I've worked with several people who've sent some samples to the soil food web analysis. There's been a few places in which I think some of their methodology may have some problems with it, as far as being able to get some good numbers. For especially, I did some analysis on mycorrhizal colonization and ended up getting some very different numbers from the Soil Food Web Lab. So, you know, I think there's some information that we can take from there and, you know, look at things like the amounts of protozoa and nematodes and bacteria to maybe get some ideas about nitrogen, potential nitrogen availability. I think there are some other labs that are coming online that are trying to do analysis on the soil food web, and they may have some better ways of looking at it. Some of it may be a little bit harder to interpret. We do things like fatty acid analysis and DNA analysis to try and identify groups of organisms. And sometimes that can be difficult to do and expensive to do, as well as a little bit difficult to interpret. And the more we're building up a database of information for agricultural fields, the Soil Food Web is located in Oregon. It's been associated mostly with the horticultural industry, which is a very different environment because usually they completely sterilize their soils. So I think that just like with soil testing, there's some misinterpretation that can be made because we don't know what the baseline should be. So the more we can get different soils analyzed, the more we can get information on what the baseline should be, so what makes things good or bad, because we don't really know that very well for a lot of agronomic systems as far as individual species and amounts needed.
Okay. All right. Thank you, Doctor. Yeah, for those, a soil food web analysis is just a measurement, a quantitative measurement of the various organisms that Dr. Nichols talked about. Again, we appreciate your participation. And just before we close out, I just want to make folks aware of two things. You can listen to a replay of this presentation at this website, and which is the New Science and Technology training library. And if you go to that, I'll just click on it real quick. This is what it looks like. You can access it there at the address that's on the webinar. Here's the last one we did. We will post this presentation there. If you hit download, you'll just simply go to a PDF file here that gives a description, what the objectives were, gives a basic description of what the training session was, and then you can either hit play or download. I would suggest you hit the download because that gives you the opportunity to pause and back up or fast forward through the presentation, where the software, if you hit the play, doesn't allow you to do that. And then the final, make sure you get your presentation or your information into us about if you have your CCA credits. Make sure you get that information either faxed or emailed to Holly Kirkendall. And then the last thing is our next MRBI presentation will be on Wednesday, and that should be August the 10th, not July 27th. That's a cut and paste error there. So again, Wednesday, August the 10th, it'll be Mark Scarpetti, who's the state agronomist in Ohio, and he's going to be building on what Dr. Nichols talked about. How can you take these principles and put them into a conservation plan and use them for the foundation for resource management, looking at planning to improve soil health, managing more by disturbing less, diversifying, growing a living root throughout the year, and keeping the soil covered. Those are the basic principles he'll be covering, and he'll be talking, just kind of expounding on that. With that, I appreciate everybody's participation. Again, Dr. Nichols, you did a great job, and I assure everybody from what the responses I'm seeing and the Q&A and the applause I'm getting over the computer here, everybody's in agreement. So again, we thank you, and I guess that we'll just call it a day. Thank you.
Thank you.
Hey, Chris, hang on for a second.
Yep.
Yeah. Boy,
One moment, please.
Okay.